Hello, I'm Fran Chiaramonte, the Program Executive for Physical Sciences at NASA headquarters for the Space, Life, and Physical Sciences Division. Today I'd like to talk to you about colloidal systems and biological macromolecule experiments on the International Space Station. Our colloidal program that we have developed is international in nature. Before the most recent NASA research announcement selections were made, the program consisted of 7 investigations, 4 of them from NASA, 2 from ESA, and 1 from South Korea. One of the U.S. investigations, as a matter of fact, is funded by a major U.S. corporation working through CASIS, also known as the Center for Advancement of Science and Space. With our NASA research announcement, we added 3 new additional investigations in colloids and 3 new investigations in macromolecular biophysics. All the investigations will use the light microscopy module on the International Space Station. For the colloidal experiments, we are building 4 sample modules: a basic module, a module with simple heating, a module with a refined temperature control, which can include a temperature gradient, and a module where we can apply electric fields. For the biology experiments, on the other hand, we are building 1 unique sample module. This module will be able to use a syringe to inject biological samples directly into the sample cell with the help of the astronauts. The samples can then be preserved by freezing them on orbit for additional study on Earth. Currently, the microscope collects 2-dimensional slices of either the colloidal or biological samples that are being studied. Ultimately, we will add confocal microscopy, which is really the prize. This will give us 3D renderings of any sample being studied. The advantage of microgravity for colloids is that the particles no longer settle or jam, but remain suspended in solution. The aim is to observe colloidal processes, to understand them, to learn how to control them using the microgravity environment of the International Space Station. These colloidal systems may be used to model atomic systems and to build nanotechnology devices. We will be able to make new structures by using self-assembly, which should lead to novel items such as nanopumps and photonic crystals for future optical switching and computing. For biological macromolecules, on the other hand, we have seen larger crystals with fewer defects when grown in microgravity. This occurs because molecular transport is diffusion controlled in space instead of being driven by natural convection as it is on Earth. Higher crystal quality will help us determine the structure and which drugs can bind with that structure. Typically, the goal is to bind the drug with the macromolecule to prevent it from doing any further damage. What I've just described is the way forward for colloidal research and the study of macromolecules using the microscope on the International Space Station. We hope to have future opportunities to select researchers such as yourselves to join us in this international program. Thank you.